Hi there and welcome to this IB biology revision video, and today we are focusing on video 4, which is amino acids and proteins. So let's get started, and I'll just remind you that this is a video that is to support you. It is not to replace full revision, and hopefully it will help you if you have any issues. So proteins then have essential structural roles and also functions, as you'll learn later on. They're particularly part of your muscle cells. But in terms of function, just to give you an idea, you'll study them in different places. You'll study about antibodies in disease, fighting off disease. You'll study about enzymes, hormones, transport proteins such as hemoglobin, structural proteins, proteins like carrier proteins that allow things in and out and across cell membranes, and contractile proteins like actin and myosin in muscle. But all of those things you need to be able to link together and realize they're all proteins. So protein, the monomer of a protein is or we should say monomer of a polypeptide is an amino acid, and you need to learn the amino acid structure. So the amino acid basically has the following key parts to it. So first of all, you have an amine group, okay, which is this NH2 bit here. And there you have a carboxylic acid group, which is this C double bond O attached to a hydroxyl group here. And in the middle, the carbon is called the alpha carbon. Now, I always tell students to learn this by thinking it as having, again, if you think about the previous videos, I said backbones. So this has got an NCC backbone, and I find if students learn that, they tend to be able to build the rest of it around it. There is also this R group, which we'll talk about in the next few minutes, which can be one of 20 different combinations of atoms, which gives the 20 different amino acids their structure. So here we see again the basic structure of amino acid, okay, and notice here they've actually called the different groups the amino group and the acid group. So as opposed to the amine group and the carboxylic acid group, you normally can shorten down the names of that. So that is a bit of an easier option there. And here you can see this is your R group, obviously normally, which can be positioned above or below the alpha carbon. And in this case, the R group is hydrogen, giving you the simplest amino acid. I would suggest you learn this one. Glycine is the simplest amino acid because it has the smallest R group. Hydrogen, one hydrogen atom, and hydrogen is the smallest atom possible. And if you look here, you can see how the different amino acids combine. So you've got one amino acid there, attached to another amino acid there, attached to another amino acid there. That's the R group of that amino acid. That's the R group of that amino acid, so they're different R groups. And then this one, the R group is hydrogen, saying that that must be glycine. So the R groups are what give the amino acids their variety. It gives the protein the variety. They all have the identical structure apart from the R group, and that gives it its characteristics, and that's obviously then going to give you different properties. And actually even some amino acids can even be further modified. So proline is sometimes modified as a hydroxyproline to increase collagen stability, so they can be further modified by having reactions happening with them, but you don't need to worry about the details of that. And just to give you some idea of the amino acids and what they have as R groups, we have hydrogen only is one amino acid, glycine. Ones that are only made up of hydrogen and carbon in their R group, there's 5 amino acids for that. Ones with sulfur, there's 2 amino acids, and so on and so on. So you can see the variety of R groups that there are in amino acids. So there are 20 amino acids in total. Plants can make all of the amino acids needed as long as there's nitrate in the soil. So this will link to something called the nitrogen cycle that you will need to learn later on. And this can be turned then into the amino group, joined to organic products, and make amino acids from photosynthesis. So photosynthesis actually does make amino acids indirectly. Animals, however, obviously need to have amino acids in their diet. And what happens is when you eat, you do hydrolysis reactions, breaking down proteins into amino acids, and then these can be rebuilt to make new proteins as your body needs. Animals can convert one amino acid into another, so we can make something called non-essential amino acids. But there are 9 of the 20 amino acids that we cannot build from body's material, so they must be part of our diet. So we need to eat them within our plant-based diet, and that is what we call the essential amino acids. Plants contain fewer essential amino acids though, so people who are just vegetarian do actually need to be very careful with their diet. And soya is an example of why. One reason soya, I think it tastes disgusting, is one of the key ingredients, is because it contains all essential amino acids. So we can get our essential amino acids from plants, from soya, and obviously animals that eat plants, they might have them in them, so that's where we again get them as well. So to give you an example about this, 9 amino acids said are essential. Animals cannot build them from our body. They must be made as part of the diet. Okay, but here's an example of an amino acid that we can make and how we do it. So you have an amino acid here that is an essential one, phenylalanine. That then can be used, you can add hydroxyl to it to make this amino acid in our body. So our bodies can do that for us. You don't need to know examples of the amino acids. Like everything though, if we eat too much, most things in our body can become toxic. So this is why we need one of the reasons we need the liver, because too many amino acids can become toxic, and we have to break those down into urea, which we later convert into urine, which you'll learn about later on in the course. And the process where you break down excess amino acids in the liver is called deamination. Just a little point here, I don't think it's absolutely essential for us now, but amino acids can act as buffers, which means they can resist changes to pH. And the reason they can do this is because they can either gain protons or release protons, or gain OH minus ions. So you see here we have a amino acid. Okay, it can gain an extra hydrogen. Here you see there, okay, and again you can gain extra OH group. But again, just to give you that basic understanding of it, you can have a look at it yourself more if you need it. It's not the point of this video. So going back to what we've done before in videos 1, 2 and 3, we always need to come back to condensation reactions and hydrolysis reactions. So if we want amino acids to combine, we have a condensation reaction. So remember what I said to you before in previous videos, and go back and watch them if you're not, you need to always be talking about condensation reactions when you are combining 2 molecules together. So here we have 2 amino acids, a bit different because there's no OH in the middle on the second amino acid here on the amine group here, okay, but because it's a different enzyme, you don't need it. We take out the water, okay, so it's a condensation reaction, and then that leaves this bond here for carbon and this bond here for nitrogen want to combine, so they naturally combine here. So how would you answer this question in an exam? Again, you've always got to be really specific. So you would say 2 amino acids combine via a condensation reaction. This releases a water molecule and that forms a peptide bond. Do not mention the word covalent, you won't get the mark for it. And the peptide bond is this bond here, and you are forming a dipeptide molecule, which is 2 amino acids combined. Now just be careful here, because what you sometimes see in exam questions is students saying it's formed a dipeptide bond. That does not exist. The thing that it forms is a peptide bond, okay, and you would not get the mark for saying a dipeptide bond. A peptide bond is what is formed between 2 amino acids to make a dipeptide molecule. So make sure you don't get those two confused, because again you wouldn't get the mark for that. Now if you want to break these down, so think about it, in digestion you might break down a dipeptide into its 2 separate amino acid monomers. You again then have to add the water back in. So in this case we are going to be doing a hydrolysis reaction to add the water back in. That breaks the peptide bond, okay, and water is used up to do that. So really essential that you constantly refer to hydrolysis and condensation reactions. And here's another example of it, just showing you how a condensation reaction forms a dipeptide, a peptide bond through when two amino acids combine. You also need to be aware of the protein test. So the protein test is a Biuret test. It reacts with the peptide bonds in proteins. You don't have to heat it. Now, I'm only mentioning that because some people get it confused with Benedict's test. And it goes rapidly into a purple or lilac or mauve colour if you get a positive result. I'm just going to highlight this bit though, because people get this wrong all the time. They will say it reacts with the amino acid. It's not. It's reacting with the peptide bond when you're explaining it. So now we go on to the higher level stuff only, and this stuff is about what we call the levels of protein structure. So standard level students do not need to continue from this point. So when we're making proteins, a protein is made of loads of amino acids joined in a long polymer called polypeptide. A protein may be one long polypeptide or several of them. For example, hemoglobin, which we'll focus on later, is 4 polypeptides. And amino acids are sometimes called amino acid residues because they've lost part of the molecule in the reaction, but I don't think you need to worry about that for the exam. So effectively there are 4 levels of protein structure, 3 of which are shown here. So when we're talking about the primary structure, we're talking about each individual amino acid and which one is attached to the next one. So we call this the sequence of amino acids. We then look at, well, what happens to them when they bind? And they don't just form a nice, neat straight line like this. They will form different shapes. So they might do a helical shape called an alpha helix. So there's an example there, example there, example there, example there. Or they might do something called the beta pleated sheet, which is that. We then take that and we further fold it. So if you imagine that this was still helpful like that, this has been further folded, okay, to make the tertiary structure, and we'll look at that in more detail in a few moments. So if you were asked to describe the structure of protein, you would start off describing the primary structure of protein, and you would talk about the fact that each protein has its own structure and the fact that it's made, and that all comes down to the amino acids to start off with. So the primary structure is the sequence of amino acids and the order of those amino acids. The best way is this bit that's underlined here, which says they are the sequence of amino acids, and it's called the primary structure. And obviously different amino acids will have these different R groups. So because of these different R groups, that will then give different properties, and that will then affect the shape of the protein later. And obviously, just to go a bit further forward, you'll link to this in the DNA lesson coming up, but what decides the order of amino acids, and you wouldn't put this in a question asking about levels of protein structure, but what decides the order of amino acids is the DNA code, which is organized into 3 letters called triplets or codons. So every 3 bases codes for 1 amino acid, and if you get a mutation, so you get this one here where it's gone from T and A to A and T, this might change the amino acid. But just remember, the primary structure is the sequence of amino acids. Now, even though we're talking about only 20 amino acids, that gives us a huge variety in structure, and that gives us the reason we have so many proteins. So here we have 4 amino acids in a chain, and what we're looking at here is this idea that amino acid 1, could be one of any 20 amino acids. That amino acid 2, could be one of any 20 amino acids, and so on and so on. Now, what that means is if you'd only had 4 amino acids together, it would be 20 potential combinations here. You'd have to multiply that by 20 here, multiply that by 20 here, multiply that by 20. So for just 4 amino acids, you would have that many different combinations of amino acid sequences from there. So 160,000 in 4 combinations. The small proteins normally contain more than 100 amino acids. The smallest protein is insulin, and that will contain about 50 amino acids to start off with. It gets a bit more complicated than that, but shows you there's a huge variety of amino acids. Now, the other thing then that we need to look at is that when the primary structure is formed, the bonds between alpha carbons and the adjacent nitrogen can be rotated. And what that means is it allows further shaping of the protein. So you'll hear this term quite often as we move forward, called the conformation or conformational shape, and that is the 3D shape of a protein. So the bonds can actually be rotated and bent to make the right shape of the protein. So we've gone past now the primary structure, which is the sequence of amino acids. The next level is the secondary structure. Now, the secondary structure in amino acids is one of two structures. It is either this spiral shape here called an alpha helix, or this sort of up-down sort of shape here called the beta pleated sheet. Now, this is caused by hydrogen bonding, and the reason it's caused by hydrogen bonding is because if you look at the amino acids and look at the groups, you have a C double bond O group. So if I just do this in a different color to show you, make sure it stands out for you. I'll do it in the sorry, I'm a bit slow myself here. I'll do it here. So we've got the C double bond O group, which is slightly negative. So what we're saying here is this bit, which I'm highlighting on that one there, is slightly negative, and down here on the beta pleat sheet is that one there. Okay, so the C double bond O is slightly negative. The C double bond, sorry, N to H bit then is slightly positive, and therefore we get this delta positive, delta negative, and as a result you get these hydrogen bonds forming between the two. Now, what decides whether it is a beta pleated sheet or an alpha helix is the amount of hydrogen bonds and the position of the hydrogen bonds, and what's going to affect this is what the amino acid is. So because of the different R groups in the amino acid, that will depend on the shape. So you have all these different R groups which alter the shape of the amino acids. We then have the tertiary structure, which is the further folding of the amino acid chain to make the polypeptide. So you could have alpha helix within this, so just point it out, it could be like that, okay, it could become beta pleated sheet, it could be alpha helix like that, but this is the further folding into the shape, that 3D characteristic shape of an enzyme, and it is determined by the primary structure and the bonding between the amino acids, and it forms one of 3 bonds. So the 3 bonds that form are ionic bonds. So you should know that ionic bonds form between a metal and a non-metal, between a positive and a negative ion from GCSE. You get disulfide bridges that only occur between the amino acid cysteine because they have sulfur in them, okay, and then you have hydrophobic and hydrophilic interactions where the protein, the polypeptide, the amino acid I should say, interacts with water as well. Now I should really say there's also hydrogen bonds obviously here as well. So the shape then of the tertiary structure develops actually during protein synthesis. So in protein synthesis you'll learn the final bit where you stick the amino acids together and make them bond, forming peptide bonds, is called translation. And during the translation process, something called a chaperone protein will then make the shape of the protein. So proteins will have a very specific shape, and the chaperone protein does that folding, causes that conformation shape, and basically the chaperone protein makes it the right shape. Now, not all amino acids effectively have a tertiary structure. If you are what we call a globular protein, that means you are highly shaped, you've got a tertiary structure, whereas fibrous ones do not. So we'll look at now, some proteins have more than one polypeptide, and if you have more than one polypeptide chain, you have a quaternary structure. and basically different chains might interlink in different ways together. So the the hemoglobin molecule has 4 polypeptide chains. We call them 2 alpha and 2 beta chains. These aren't linked to alpha or beta pleated sheets, they're just names given to these chains. And those 4 chains link together, and they have an extra, what we call prosthetic group, which is a non-protein part of the structure, which is made of iron, as in the metal iron. We call this the heme group, and it's an ion because it's got a positive charge. And that's the bit that actually does the job of holding onto oxygen in hemoglobin. So we need to just be aware that there are 2 different groups of proteins then called conjugated and non-conjugated. So a non-conjugated protein is something like hemoglobin. So just mentioned the iron group in there, which acts as the heme, and it accepts the oxygen. So a conjugated protein has multiple non-polypeptide subunits, so bits that are not amino acids. In this case, it is the hemoglobin, has the 4 heme groups. As I said to you before, that is the iron. So it is Fe2+, I believe it is, okay, and that is what then accepts the oxygen molecule. Many enzymes have this non-polypeptide subunit, so it's essential in there. An example of a protein that would need it is also chlorophyll, which has magnesium in the centre. Now, an example then of a non-conjugated protein would be insulin. So this is something that doesn't have any extra non-polypeptide groups, and it's linked by interactions between the tertiary structure. So you see here there are 2 chains in insulin, 2 amino acid chains. They both got their own tertiary structure, and they are linked together due to interactions between those tertiary structures. There's no iron in the middle of those. And then when we go back to what we mentioned a few minutes earlier, there are 2 what we'd say main groups of proteins, whether they have a tertiary structure or not. They are globular proteins, and then there is fibrous or fibrous proteins. So globular proteins are spherical shapes generally. They are used in hormones, in enzymes, and they have biochemical functions as a result. They often have a very 3D shape, a very specific shape that, for example, might have an active site if it's an enzyme. They have a huge variety of shape, and they can be denatured because that shape is essential for their role. So often, if you're doing A-level biology, one of the essay questions you get is describe why shape is important. The shape of globular proteins is essential because the enzyme's active site won't work if you don't have a particular shape. The shape of a hormone is essential because it has to attach to a receptor on the membrane of a cell, and if it doesn't attach, it won't activate the reactions it needs. Whereas opposite to this is something called a fibrous protein, which is long and thin. It's got structural functions such as keratin, which makes hair, collagen, which makes your tendons, cartilage, which makes like your nose and ears. And as a result, it just needs to be, because it's structural, it just needs to be strong, it just needs to be insoluble, which means it won't dissolve in water. It's often repeating amino acids, often a lot of glycine because it's the smallest amino acid, so it can be as compact as possible, and it's often pH and temperature resistant. And you might be asked to compare the difference of the 2. So this is in your textbook, and it is how they recommend it. They recommend you use the word spades. Okay, and the word spades is to show you that you've got shape. Okay, so you need to know the difference in shape between the 2 of them. So I said one fibrous is long and narrow, whereas globular is round and spherical. Purpose, well, the fibrous has structural purpose, globular is functional purpose. Acid sequence, so the repetitive amino acids in fibrous, irregular amino acids, so lots of variety in globular. Durability, so they've got less sensitive or more sensitive. You need some examples, and you need to talk about solubility. So fibrous is insoluble in water, whereas globular is soluble in water. What do we mean by soluble for globular? Well, what we mean is it will interact. It doesn't necessarily mean it's going to completely dissolve in water. It means that it will interact with the water. It will bond with the water molecules to allow the water molecules to to allow it to react and basically use water for different things. Okay, so there it is. So quite a lot of information there. Proteins, really important, links to so much other parts of the course, and now you should be able to explain the relationship to the amino acid sequence and the diversity, so the primary sequence and how that links to the diversity of all the different protein types that we have. Be aware you might have to apply this to examples, so it is worth learning these examples or a couple of these in case they ask you to explain it, and then all you're doing is describing a fibrous protein, so you're still describing the primary structure, the secondary structure, and then explaining this bit about the fibrous to that. If you're asked to describe the shape of hemoglobin, you should know it's globular, and you should know that extra bit about the heme group and the 4 polypeptide chains that make hemoglobin. Thank you.